Hey everyone, and welcome to The Explainer. So today we're going to be doing something a little bit different. We're actually zooming all the way out from the macro medicine that we're used to, pills and standard surgeries, and we are shrinking way, way down into the microscopic world of cellular engineering. We're going to look at how manipulating matter at the literal scale of single atoms is completely rewriting the rules of human health as we know it.
Here is our roadmap for today's journey. We'll quickly cover the nanomedicine revolution today, then the CRISPR delivery problem, meet the microscopic vehicles, dive into nanotheranostics, look at transforming disease treatment, and finally, wrap up with the limitless future.
All right, let's jump straight into Section 1, the nanomedicine revolution today. Okay, so manipulating materials at the molecular and atomic level, that is honestly no longer science fiction. Just take a look at these 5 key medical applications already making huge waves in 2025. We've got targeted drug delivery. So instead of flooding the entire body with highly toxic chemotherapy, nanoparticles are now dropping payloads directly onto cancerous tumors. It's incredibly precise. Then we have nanotech-enabled sensors detecting Alzheimer's biomarkers literally before symptoms even appear. We're also seeing nanoscale scaffolds actually guiding nerve regeneration, smart implants that adjust to your body's inflammation in real time, and antibacterial nanomaterials laced with silver and copper actively fighting off those super stubborn, antibiotic-resistant hospital infections. The stakes here are just massive, and the real-world impact is happening right now.
Moving right along to Section 2, the CRISPR delivery problem.
Now, I really want you to think about this question for a second. We already have the ultimate genetic editor, CRISPR-Cas9. So why is it so incredibly hard to actually use in a living human? I mean, we have the machinery, we can program it to cut and fix faulty DNA. But the absolute biggest obstacle we face is safely delivering that microscopic machinery exactly where it needs to go, without the human body completely destroying it first. So the real bottleneck is how we transport this genetic editor. Well, the old way of doing this was using viral vectors. You basically hollow out a virus and stuff it with CRISPR. And while viruses are, great at infecting cells, they're inherently super risky. They trigger our immune system, they have a tiny little cargo capacity, and there's a risk of accidental genetic integration. So what's the definitive solution? Non-viral nanoparticles. These are infinitely safer, you can completely customize their physical characteristics, and they essentially fly right under the radar of our immune system. It's a total game changer.
Okay, let's dive into exactly what these are in Section 3, Meet the Microscopic Vehicles. I like to think of these nanocarriers as highly sophisticated, microscopic Trojan horses navigating your bloodstream. And we basically have a fleet of 4 different vehicles here, each solving a really unique transportation challenge. First up, we've got lipid-based nanoparticles, or LNPs. These are soft little fat vesicles, and they're the absolute rock stars right now, famously proven in the mRNA COVID-19 vaccines. They brilliantly encapsulate and protect that fragile genetic cargo. Second are polymeric nanoparticles. Engineered from polymers, these are kind of the customizable, slow-release capsules of the nanoworld. Third, inorganic nanoparticles. Made from stuff like gold, silica, or iron, they're physically robust and incredibly useful for diagnostic imaging. And finally, biomimetic systems. Now, these are arguably the stealthiest. They're literally wrapped in the cell membranes of red blood cells to perfectly mimic biology and completely avoid immune detection. Incredible, right? And this brilliantly illustrates exactly how this works in practice. We can literally follow the visual flow of a lipid nanoparticle carrying its CRISPR cargo, moving from formulation all the way down into a target cell. Let's walk through this process step by step. First, scientists formulate the lipid core and encapsulate the delicate CRISPR machinery inside. Then, it's administered into the body. But step 4 is where the true magic happens: cellular uptake and endosomal escape. See, when a cell swallows our microscopic vehicle, it traps it in a highly acidic bubble called an endosome. Think of it like a tiny cellular stomach designed to digest invaders. If our vehicle gets digested, well, the therapy fails entirely. But these smart vehicles use things like the proton sponge effect. They absorb the acid, swell up, and deliberately burst the endosome. They escape safely into the cell cytoplasm, where they can finally enter the nucleus and perform the gene editing in step 5. You guys, it is literally a microscopic prison break happening inside your cells. Which perfectly sets us up for Section 4, nanotheranostics, diagnose, and treat. We are entering a major paradigm shift here, where we don't just deliver drugs anymore. We actually monitor the targeted treatment in real time. Nanotheranostics is basically the ultimate 2 birds, 1 stone multitasker.
By combining diagnostic imaging and therapy into one single microscopic system, we are completely throwing out the old test, wait, then treat model. The molecular understanding of the disease and the treatment itself are customized on the fly to a patient's exact genetic profile. So, to put that into perspective, here's how this works in practice. Let's say a patient has cancer. Doctors can now inject metallic nanoparticles, like iron oxide or gold. These particles have this natural tendency to accumulate in the irregular blood vessels of a tumor, and because they're highly dense metals, they light up brilliantly on an MRI or CT scan, letting doctors visually see the exact boundaries of the tumor. But remember, this is a multitasker. Once that tumor is mapped out, a doctor can shine a specific near-infrared laser right at the area. The gold nanoparticles absorb that light, they heat up intensely, and completely destroy the malignant cells from the inside out. Bam! Diagnose and treat simultaneously. All right, let's look at the bigger picture in Section 5, transforming disease treatment.
So, how does all this microscopic engineering actually translate into curing massive macro-level human diseases? Notice the sheer breadth of medical fields we are targeting here, from cancer therapy to infectious diseases, all the way to neurological and autoimmune conditions. By pairing CRISPR with nanotechnology, creating what's called nano-CRISPR, we are launching a multi-pronged attack on some of the most stubborn diseases known to humanity. I mean, let me give you some specific, jaw-dropping examples of what this looks like today. For infectious diseases like HIV, nano-CRISPR is being used to literally snip out and eliminate the integrated proviral DNA. That's the viral code hiding deep inside host cells. During COVID-19, nanoparticles delivered RNA-targeting CRISPR to physically break down the genome of the SARS-CoV-2 virus right in respiratory cells. Over in the brain, scientists are designing nanoparticles that can actually cross the ultra-strict blood-brain barrier to edit the genes responsible for forming the plaques that cause Alzheimer's. And for autoimmune diseases, these smart particles are being programmed to target specific immune regulators, like dendritic cells, to stop the body from attacking itself in conditions like type 1 diabetes. We aren't just managing symptoms anymore, folks. We are going straight to the source code and rewriting it.
Now, what's really interesting about this next slide is where this all heads next. Section 6, the limitless future. This part absolutely blows my mind, the futurology of human longevity. As noted in discussions surrounding futurist Ray Kurzweil, there is a serious, mathematically projected possibility of utilizing nanotechnology to not just pause, but actually reverse aging. This concept is tied to something called longevity escape velocity, or LEV. The whole idea is that once nanomedicine advances to a point where we are adding more than 1 year of life expectancy to a human for every 1 year that passes, we effectively outpace aging. We reach a point where microscopic fleets of nanobots could constantly repair cellular damage, clear out senescent cells, which are basically dead, zombie cells that cause aging, and edit our genes to maintain peak biological health indefinitely.
And that leaves us right here. As you've seen today, manipulating atoms is delivering incredibly targeted cures, sneaking genetic editors past our own cellular defenses, and allowing us to diagnose and destroy tumors at the exact same time. Nanotechnology is clearly no longer science fiction. It is for sure the very foundation of modern medicine. The only question remaining is, as these microscopic vehicles become more and more advanced, just how long will they extend the human story? Thank you so much for hanging out with me on this explainer. Keep asking questions, stay curious, and I'll catch you next time.